Hi, my name's Izaak Wilkinson. I was working with Dr. Celia Blanco and Dr. Ricardo Cabrera on the Blue Marble Science YSP this summer, and I was examining the evolution of genetic code. Genetic code itself is the universal language here on Earth. It is the same for all life that we have come across. And what it specifically examines for us is the transfer of genetic information, which is the central dogma. As astrobiologists, I do want to bring us all back to Bio 101 here for a second before I get into the research side of things, just to go over some general DNA and genetic info. So the central dogma itself says that after DNA is replicated, it is transcribed into RNA, and RNA is then translated into proteins. And as you can see here in the graphic, for the proteins, they are made up of little amino acids. And all life on Earth uses a canonical set of 20 amino acids. And I will get into what I mean by a canonical set here in a little bit. But this is going to be a genetic code table where it's showing specific codon triplets and how they're corresponding to each amino acid. What a codon triplet is itself is just each of those little base pairs that we saw in the RNA. Three of them together make up a codon triplet, which then corresponds to the amino acid. And something that's pretty easy to see on a chart like this is that multiple codons are coding for the same kind of amino acids. This isn't random, this isn't by chance. These similar codons have similar properties. So this is showing us that the creation of the genetic code and amino acids were most likely shaped by some sort of evolutionary process. And one more little piece of background, I just want to give a brief overview of these canonical amino acids that I was talking about. There are 20 of them, and as you can see here, this is just giving you a little basic breakdown of their structure, as well as their name and their codon triplets down at the bottom. But the first set, the alphabetic ones here in red, form hydrophobic cores, stabilizing protein structures through nonpolar interactions. The next set would be the aromatics that you can see here in green, which provide stacking interactions and often contribute to ligand binding or structural rigidity. Next is going to be the acidic group, which carry negative charges, which is essential for electrostatic interaction as well as catalytic chemistry. Next would be the basic ones, which have a positive charge, which is crucial for binding to nucleic acids and active site chemistry. Next, we get into the hydroxylic group, which enable hydrogen bonding and serve as sites for post-translational modifications. Next is the sulfur-containing, which provide redox activity and covalent crosslinks. For example, in something like cysteine, this would be the disulfide bonds that stabilize the tertiary structure. And then finally, the amidic group is they stabilize the protein surfaces through hydrogen bonding and polar interactions. So that is kind of a general overview of the canonical amino acids and how they transfer that genetic information. And from there, I conducted an extensive literary review on the theories surrounding why these specific set of 20 amino acids were selected out of the many available on a prebiotic Earth, along with the theories of how the amino acids themselves became more diverse. So first, why the canonical 20 amino acids? There's a couple of ways that we can study this, but the most crucial thing for an amino acid to become a protein is its ability to fold. If it can't fold, it cannot become a protein. So in order to test this, we first examined reduced alphabets, which sounds a lot more complex than it is. All a reduced alphabet is taking a singular amino acid to act as a representative for a group that it's very chemically similar to. So you don't have to test quite all of them. And basically, what we found is there are a set of 10 amino acids that were prebiotically available on Earth that also support alpha helices and beta sheets, which are very important in getting a secondary structure started. But we also found that they were unable to encode for small proteins on their own. This prebiotic set of 10 would be alanine, aspartic acid, glutamic acid, glycine, isoleucine, proline, serine, threonine, and valine. So the fact that these are able to begin to make a more complex secondary structure does show that they were prebiotically available and they should be a part of the genetic code. But the fact that they aren't able to encode for these small, simple proteins means there's more diversification that has to occur with the genetic code. So that gets us into these peptide libraries that you're seeing at the side. So why with these canonical set of 20, why were they picked? As I mentioned, there were tons available on Earth, so there had to be something special about this specific set. Yet again, we are going to be examining their foldability. And for these peptide libraries, I'll break it down a little bit. 19F is going to be the full peptide library excluding cysteine. 10E is going to be the earliest prebiotic set that I was just discussing from the reduced alphabets experimentation. 10U is where we start to get into some of our experimental groups. This is going to be our unbranched alternatives where we are using norvaline, norleucine, and 2-aminobutyric acid as amino acids in to see if they can also allow for folding. 11R is going to be the same as that 10E group, but it's going to add in arginine to start to show the expansion of the code. The 11D group is going to be substituting in 2,4 diaminobutyric acid, acting as an early catatonic amino acid. And then finally, 11Y, we add in triazine to show how the later aromatics will be acting. And basically, from taking all of these libraries and seeing how they fold and interact with each other, what we're seeing is the unbranched amino acids from group 10U, as well as the short chain basic amino acids from group 11D, actually oversolubilize the polypeptides by stabilizing their unfold conformations, which was devastating for the proteins because proteins need to be soluble in order to fold. But if they're too soluble, they don't make a stable structure. But what we also saw was 11R and 11Y libraries mildly increased the secondary structure propensity when comparing it to the 10E group, showing that it does make sense that these other amino acids that we do see in the code were then added in to help make a more stable core. So that then gets us into some of the theories of expansion. There are three main theories that go over how the genetic code in amino acids expanded, and that would be the stereochemical theory, the coevolution theory, and the four-column theory. Now, I will say none of these theories fully agree with each other. So in examining these three, we have to take the strongest evidence from each one and create more of a fully cohesive story. But basically, what they do agree on is that complex amino acids formed later. And when it comes to things like the stereochemical theory, that was examining the specific chemical and physical interactions between those codon triplets and the amino acid itself, which would most likely be the earliest set to come about. The coevolution showed that after that first set of amino acids, they were able to biosynthetically evolve a next set of amino acids based off of those ones into the four-column theory of evolution, which shows when the code is evolving, focusing on the second codon in sequence, we can have much more diverse and complex amino acids while also minimalizing translation errors. So to kind of put that together to show here, this would be when we take what each of those main three theories have as their strongest points of view in evidence. This would be the order of the amino acid evolution, which does not fully agree with what we know from the prebiotic experiments. But what we're seeing here is glycine, alanine, aspartic acid, glutamic acid, and valine all have very strong stereochemical evidence. But at the same time, each of these can also have evidence showing that they can subdivide and become a different kind of amino acid, which would be things like serine, threonine, proline, leucine, isoleucine, asparagine, glutamine, lysine, and arginine. And that's going to really be agreeing with the coevolution theory, which will then lead to, through post-translational modifications, the phenylalanine, triazine, tryptophan, cysteine, methionine, and histidine. And why does this matter going forward? What we know on Earth, like I said at the very beginning of this presentation, is all life uses these canonical amino acids. And not only that, all life also uses the central dogma to explain the flow of genetic information. We would be able to assume that if life exists extraterrestrially, it would probably also follow that same central dogma as all life on Earth does, but it could potentially follow it with different amino acids. Things like solubility and salinity content is what made it so easy for some of these earlier amino acids to be able to fold and then expand into a more diverse code. So understanding the environments and processes on exoplanets can start to give us hints about how life could be evolving or just getting started off there. I'd like to thank Dr. Cecilia Blanco and Dr. Ricardo Cabrera for supporting me through this research project, as well as my friends and family who have listened to me talk about this endlessly over the course of the summer. Any questions?
Great job, Izaak. I think we have time for some questions here. Yes, Sanjay has a question.
Yeah.
Izaak, a very interesting presentation. So you made it clear that folding is necessary for the amino acids to do things and drive their evolution. Yeah. But what controls the folding itself? Is it changes in environmental conditions? Like, why do they fold in the first place and how?
Yeah, so that is definitely very related to environmental conditions. And I don't want to get too much into this because my partner from this, SM, will be presenting a little more specific details onto that. But basically, the environment of a prebiotic Earth and the theories of proteogenesis with the specific salinity contents and the pressure systems around them, the interactions of the amino acids were a lot easier.